Hello! In this video, I'm going to show you how to do gene analysis in the shell. First, I'm going to create an empty repository. I will use my template new project. I will name this repository gene analysis and set it to public. I'm going to copy this repository into my instance by using the git clone command and the URL. I will change directory to data and then download hemoglobin subunit alpha 1. To do so, I will go to the NCBI browser and search for hemoglobin alpha 1. We can click on the GenBank link and it will take us to the GenBank file. This contains information for the entire human chromosome 16. I will copy the reference sequence number and use the command ncbi HCC download. I will download the GenBank, FASTA, and GFF formats. We can see how this looks. The FASTA format contains the nucleotide sequence of chromosome 16. The GenBank format contains different features such as genes, exons, mRNA, etc. Finally, the GFF file also contains genomic features stored in a table format. We can see that this gene, hemoglobin subunit alpha 1, has a gene ID 3039. We can use grep to filter the lines in the GFF of chromosome 16 to select only the lines that contain gene ID 3039 and save them into a file called log3039. We can use less to open the file. This gene may contain different variants produced from alternative splicing. If we use the flag V and then add X2, we can remove all the variants called X2 from the file. Then we will use the program genome tools to convert this back to the GFF format. We can use the program genome tools stat to count the gene length, exon length, intron length, CDS length, etc. for our file. Here you can see that this contains one gene, one protein coding gene, one mRNA, 3 exons, 3 CDS, and 2 introns. I can also do the same analysis with the entire chromosome if I don't filter using grep. Here I can use GT sort for the entire chromosome, and then GT stat will produce the gene length, exon length, intron length, and CDS length. We can open the output using less, and we'll see that this chromosome contains 468 genes. 855 of them are protein coding genes. It also contains 484 pseudogenes. We can also filter all the lines that contains the word CDS in our file for hemoglobin. This will save only the lines for coding sequences that correspond to the 3 exons of this gene. Then we can use the command bedtools get FASTA to obtain the sequence from the FASTA file using the coordinates in the GFF file. I will also use the function union from the program emboss to concatenate 3 exons into a new file called CDS union. I will use transseq, also from emboss, to translate the nucleotide sequence into amino acid sequence. Here is the output of the nucleotide sequence, and here is the output for the amino acid sequence. Notice how this sequence starts with methionine and ends with the stop codon, symbolized by start.